I'm Ed Bynum, and I'm a retired extension entomologist. For the first presentation, I'm going to be talking about the release of predatory mites for managing spider mites in corn. With that, we need to identify the different species of spider mites that we have on the Texas High Plains. Our primary species is a Banks grass mite. It's the most common and it usually occurs early in the season in comparison to the other spider mite, which is a two-spotted spider mite, which occurs a little later in the season. It can occur anytime throughout the season. For the Banks grass mite, it generally starts on the lower portion of the plant, on the lower leaves. And then as the populations build up, they begin to desiccate the leaves and they move up the plant. But oftentimes the two-spotted spider mites, they can be found on almost a majority of the leaves all the way up and down the plant. Now the two-spotted spider mite is a little harder to control with most of the acaricides that we have in comparison to the Banks grass mite. But both of these species now over time has developed resistance to a lot of our acaricides that we have available to control our different mite species. Again, the spider mites, they cause the damage to the lower part of the plant as they're feeding. And as they feed, they have two little styluses that punctures each individual cell. And as you have the populations build up, then they can kill and cause quite a bit of damage. So by the feeding, it lowers the chlorophyll content, which actually reduces the transpiration and cooling and also affects the photosynthesis. So you don't get the growth and the buildup of the yield within the plant. For corn silage, we have seen through studies that we can have about 17% loss in our tonnage. For grain, we can expect yield losses of 23% in corn and cotton losses of 67%. So heavily infested plants are also prone to lodge. And the grain corn, there's not any more damage once the corn is in the dent growth stage. Will not be any more yield loss for the grain yield. This slide just shows the level of damage that we can get from our spider mites. As you can see with this leaf, that speckling yellow part is where they have killed the individual cells on the plant. And then on the left side, you can see the desiccation on the lower portion of the plant. And this is what reduces our yield. We typically rely on predators to help us manage the spider mite infestations. Unfortunately, the spider mites will build up populations much faster and quicker than our predators will build up their populations. But as the populations build up for our predators, they will eventually control the spider mites. And we have 4 different predators that we rely on during the season. And they generally show up a little later in the season. And that would be the predatory mite, the minute pirate bug, 6-spotted thrips, and the spider mite destroyer. With this particular slide, you can see which adult or nymphs or larvae, how many spider mites they can consume within an hour. And they all can do a real good job in controlling and consuming spider mites. And so we kind of rely on all 4 of them to help us to be able to manage the spider mites. This work was done by Dr. Wilson and Dr. Archer back in the 1990s. But with this test, we also evaluated predatory mite to see if we could be able to make releases of the predatory mite to be able to manage the spider mites. At this time, when we did this study, we were able to manage the spider mites when we made releases of the the predatory mite at tassel. But unfortunately, it was too cost prohibitive to be able to make these releases. But we knew that we would be able to make releases to be able to manage the spider mites. So when I set up the trial to try to evaluate early releases of the predatory mites, I selected 2 different predatory mite species to use. One of them is the Glandromus occidentalis. This predatory mite does well under really hot, dry conditions. And since we can have those conditions on the Texas High Plains, I thought this might be a good species to be able to use. The other mite species that we used was the Neoscilus fallacius. This is a native predatory mite that we have on the Texas High Plains, but generally the populations don't show up until later in the seasons. So by using this native species, it generally is a good fit for us in our situation. So I thought this would be good to be able to make releases earlier in the season. When we were setting up the test, we had contact with a crop consultant, Mr. Olin Moore, and he got us in contact with one of his producers in Castro County near Dimmitt. And so he allowed us to be able to set up our test plots in his fields. Picture on the left shows that we had 4 different fields to choose from. We used field 1 and field 3 in 2016, and we used field 1 and field 4 in 2017 to be able to set up our experiment. The picture on the right shows how we set up our plots so that we could be able to determine whether predatory mites were controlling the mites in comparison to when we did not have any predatory mites in certain plots. The P1 represents where we made releases of the predatory mites. P2, we made releases of the predatory mites a little further into the field. P3, we did not make any releases of the predatory mites, but it gave me an indication of whether or not the predatory mites was moving throughout the field. The A1 and A2, we made applications of Asana, which is a pyrethroid insecticide. It kills off predators itself, but it does not have any impact on spider mite populations. So we could make comparisons of what our predatory mites were doing in comparison to where we did not have any predators within our plots. So we began looking for the spider mites the first week in June. We set up plots by June 15th, and we began to make counts within our plots. We had the predatory mites delivered to us at a Texas A&M Research and Extension Center, and we made these releases in these designated areas by June the 20th. Within 1 to 2 days before and after predatory mite release, those plots that we designated to be sprayed with Asana was put out, and so we did remove those predators within those plots. We did continue to count the spider mites and looking for predators within that field throughout the season until the end of August. The predatory mites that we received were from our supplier, Rincon Vitova Insectaries. And again, we had 2 different species of the predatory mites. And the Amblyseius fallacius, it overwinters in cold and late season. Gallindromus occidentalis, it does prefer warm weather and it tolerates really dry conditions. What we did, we released 8,000 predatory mites for each species in each field, which means we had 2,000 within a small container for each of the predatory mite species. We had 2 sites in each field, and we had 2 locations within the field. And we had a total of 16 containers, 8 containers for each field. And so what we wound up putting out was about 16,000 predatory mites. Now, the cost of the predatory mites, when you purchase a container that has 2,000, it costs $26 per container. Since we had 16 containers, 8 containers for each species, it came to a total of $416, and that was for both 2016 and 2017. The packing fee was $6.50 per box by 2 boxes, for one for each different species. And our shipping cost was between $174 and $165. And so our total for 2 fields was $603 in 2016, roughly $594 for 2017. And so you divide that by both fields, we have $301 and $278. So each field was 120 acres area, but we only treated those 2 hotspots areas. And so it was a cost of $2.51 per acre in 2016 and 2017, $2.48. In 2017, we began to wonder if we really had got good release of the predatory mites. And so I ordered another shipment of predatory mites a week later, and that added another $1.20, $1.70 per acre for that field. That still is a relatively cheap expense for trying to manage spider mites. This is what the little bottles look like with the different species. And there was 2,000 predatory mites in each one of those little bottles. What we would do, we selected a row. One was for one species and the other row for another species. And we would walk about 4 miles an hour and just gently shaking out the contents out of that little block. We would be able to walk about 300 feet, making the releases of the predatory mites. So we got them spread out within that test area. Now let's look at an example of our data using results from 2016. What these graphs show is the increase of the spider mites, what type of damage levels that we had, the predatory mites that we recovered during the field, and a total number of predators. This is an average for both locations within one field. So when you start looking at the spider mite populations, when we had made the releases on June 27th, the populations were extremely low, as you can see. And then by July 29th, the populations began to increase. They increased in our Asana plots, fairly heavy. And by July the 25th, in the P3 area where we did not release the predatory mites, we had a hot spot there where we had some spider mites starting to build up. And they built up fairly heavily. But then the populations began to drop off after that by August. And so looking at the damage levels, we can see that for the Asana plots, A1 and A2, the red bar and the yellow bar, we had much higher levels of damage in those plots than what we did where we made the releases of the predatory mite. And eventually in that P3 area where we did not release the predatory mites. So when you look at that, we were able to reduce the amount of damage that was caused from the spider mites. Looking at the predatory mites graph, you can see on June 27th, we began to see predatory mites show up in our area where we made releases in that predatory 2 area. And Then we began to see some predatory mites show up in our sauna area where we had sprayed with a sauna to try to keep the predator out. But yet those predatory mites, they were moving around in that field, and they were moving into those areas where we had sprayed. As the populations build up over time, you can see the number of predatory mites built up to pretty heavy levels in some of those plots. They built up really heavy in the sauna plots, and that just shows you that the predatory mites are finding and reproducing and building up in areas where you have really heavy spider mite populations. But where we had released the predatory mites in P1 and P2, we may not have had as heavy of the predatory mites in those areas, but the predatory mites were keeping that population down. And so because it was keeping the population down, we didn't see that spike in the spider mite population. The last part of this slide we're showing on total predators. And on June the 27th these are other predators besides just the predatory mites. They were primarily in June the 27th, they were primarily the western flower thrips. And we had a situation in both fields in 2016 and 2017 where western flower thrips began to migrate out of our maturing wheat fields, and they were moving into our corn plots. And when they were moving into our corn plots, they were also causing damage to the plant, but they were also feeding on the spider mites. And so that's one of the other reasons why we started out with low populations of the spider mites during the season in our test area. So when they did that, we delayed getting the predatory mites by a week so that we wasn't making the releases of the predatory mites when there were also a lot of western flower thrips out there. The western flower thrips finally dispersed, and so you can see we had a reduction in the number of predators. And later in the season, the majority of the predators began to be predatory mites. And then later on in the season, as typically we get a lot of our other mite predators building up within our plots. And so we felt very good that our predatory mites were helping manage our spider mite populations earlier in the season. So we were very comfortable and we feel very confident that the predatory mites are going to be able to manage spider mites in our population, so we can use the predatory mites, make releases earlier in the season. So in summary, our spider mite populations were very low across the high plains in 2016 and 2017. And that was primarily because of the heavy migration of the western flower thrips earlier in June. But later in the season, they did build up to damaging levels. Now, during the season, area crop consultants and that were scouting other fields, they did not find any predatory mites in their fields until the very last of August and the early part of September. So because we had made releases of the predatory mites in the fields that we had, we were able to get the predatory mites established much earlier in the season. And after our releases in June, they did establish, and we really had very low levels of spider mites to even begin with. So that is encouraging that even though spider mite populations could be low, you could go ahead and get the predatory mites established. And during the period during while we were counting, after we make our counts, we'd go around and inspect other parts of the field. And a couple of weeks, 3 weeks afterwards, we were able to find predatory mites throughout the entire field. So that was very encouraging to us. And we believe that the predatory mites was a key factor in preventing the spider mite populations from developing to high levels and causing heavy damage across the entire field. So what are some things that you need to consider when you're going to be looking at getting predatory mites and making the releases? First thing that you need to do is contact the supplier before the season ever starts so that you can arrange shipments of the predatory mite species and have an estimate about the number of predatory mites that you're going to want to make releases with and what timeline you're going to be looking at getting the predatory mites delivered. Because it takes time for the supplier to build up those populations for you. So begin scouting and marking hot spots in the outer edge of the field. So those would be the places where you would be making the releases of the predatory mites. And depending on the number and location of the hot spots, then you can make a decision on the number of predatory mite bottles that you're going to want to make releases. And of course, that's going to be dependent on the cost of the predatory mites. So contact the predatory mite supplier again and confirm the number of bottles that you want to be shipped. And then watch closely the spider mite hotspots. And if you begin to see western flower thrips migrating in to the field from as the wheat dries down, then you may want to delay the predatory mite shipments for 7 to 14 days to give time for the western flower thrips populations to decline and for the populations to spread out and not be so concentrated on the corn plants. Then recheck the spider mite hotspots to see if the spider mite colonies are still active with fresh eggs and immature mites. And if you need to, you may have to find some other areas where you do still have some hot spots. Also, you need to contact the predatory mite supplier then to confirm the actual shipping date that you want. And remember, only schedule the number of bottles for a field that you can release in the morning. You do not want to make these releases later in the day when it gets to be really hot. So a day before you going to get the shipment, kind of prepare. Have some large enough ice coolers that you can keep those shipping containers of the predatory mites in a place where you can keep them cool. And then have a plan on how you're going to make the releases in a particular field. How many sites that you're going to be making releases, the hot spots. And what we found out when we made the releases, it was going to take between 5 to 15 minutes to empty a bottle. So because of the time frame, then that can help you determine how long it's going to take you to make a release within a field. And the time that you're going to be traveling from one field to the next, so you can make the determine on the number of bottles that you're going to, number of fields that you can get predatory mites released in the morning. And at the release site, mark the starting row of where the predatory mites were released. Follow the instructions that the supplier will give you about how to make releases. And for most situations, releasing predatory mites by hand is an efficient method to be able to do that. As an example, the crop consultants that we were working with on the fields that we made these releases and the test plots, after we finished the test in 2017, he began to make releases of the predatory mites in 2018 and 2019 himself. He would started out purchasing a device that the supplier had suggested that he used to be able to make releases, but in 2019 he went back and just started making releases by shaking out the bottles with the predatory mites. So gently shake the predatory mite bottles over the corn row and you can walk approximately about 4 miles an hour, and you can empty that complete deal. What I would do after you emptied it, go ahead and leave that bottle in maybe at the base of the plant or in the whorl, because there may still be some predatory mites walking around within that bottle and in the lid. So with that, I think you would be able to get good control. Again, it may take 7 to 10 days before predatory mites can be found after you made the releases. But hopefully you can be able to see that in a very short period of time after that. And so with that, I want to thank you for the opportunity to be able to share about making predatory mite releases. And so this may be a better alternative than maybe some of the acaricides that you have available to use. Thank you.